Hello friends. In our previous period, we discussed about the endoplasmic reticulum. Endoplasmic reticulum is the extensive network within the cytoplasm of the cell of three-dimensional interconnected membrane-bound channels. Endoplasmic reticulum carries some vital functions of the cell. It is associated in the protein synthesis, in the lipid synthesis, transportation, storage of the proteins, so many secretory products, secretion, and at the same time it is the membrane factory. It supplies membrane wherever needed. The membrane is needed by the membrane bound cell organelles like lysosomes, Golgi complex, plasma membrane or the cell membrane, nuclear membrane, and like that. Now this endoplasmic reticulum is a part of endomembrane system. Endomembrane system is a system in which there are few cell organelles where there is coordination in the work and where there is membrane flow, GERL system. Golgi, endoplasmic reticulum, lysosome system. Many functions, many activities, many processes are conducted by these organelles with coordination. They coordinate with one another. So when we talk about endoplasmic reticulum, naturally we have to talk about the Golgi complex, and that is our today's topic, Golgi complex. Let's discuss about the Golgi complex. It is Golgi complex. It is also called as Golgi bodies, only Golgi dictyosomes in the plants. It is called as dictyosomes in the plants. So this Golgi complex is equally important cell organelle. It was discovered by Camillo Golgi, and that's why the name Golgi complex. In 1898, and he got Nobel Prize for this work, Camillo Golgi. He is a Nobel laureate. So this Golgi complex, which was discovered by this man, is very important cell organelle because it is the middleman of the cell. It is export house of the cell. Golgi complex is very fascinating cell organelle. It is called as middleman of the cell. It is called as export house. It is called as shipping center. Golgi complex is called as shipping center. It is the center of export is there. It is also the center of manufacturing. It is processing, refinery center. A refinery center of the cell. It is also called as dictyosome in the plant cells, middleman, export house of the plant cell, distribution center of the plant cell, shipping center, middleman, or a refinery. Processing unit. It is also the processing unit. See, all these terms are coming by its function, by its work. So this is one of the most important cell organelle, and that's why it is called as middleman. It is called as export house, shipping center, refinery center, processing center of the cell, dictyosomes. Why? Because it is distributing the material. Whatever synthesized by the endoplasmic reticulum, that is also distributed by the Golgi complex itself. Golgi complex synthesizes some materials by its own, but the materials which are synthesized by other cell organelles like the nucleus or the endoplasmic reticulum, that material is also sent to its destination by this Golgi complex, and that's why this Golgi complex has so many names. Now it is called as dictyosomes in the plants. In plants it is not interconnected, the lamellae or the cisternae are not interconnected. Just like endoplasmic reticulum, it is membrane bound and it is made up of some cisternae, vesicles and the tubules. So it is present in three forms like the endoplasmic reticulum. The forms are cisternae. These are the flat membrane bound stacks. They are arranged down one to another, and generally 4 to 8 stacks are present in one Golgi complex. Vast number of Golgi bodies are present in each of the cell, especially the secreting cells, the cells which are synthesizing some materials and secreting them. In those cells they are very large, up to 600 Golgi bodies have been counted in some cells. So their number is quite large. The forms are different. In case of the fungi, the cisternae are single. Golgi body is represented by single cisternae. They are not forming the stacks. But in many cases, you can see here in the figure, the stacks are present. In each stack, 4 to 8 cisternae are present. These are the membrane bound sac-like structures with some lumen inside it, not like the endoplasmic reticulum. Endoplasmic reticulum encloses a huge space of the cell, but here the space is limited. Though it is limited, it is definitely present there. Then the tubules are present, but they are less in number, and the vesicles. Even vacuoles, some large vesicles or vacuoles are also present in the Golgi complex. Now, in many cases, the cisternae are interconnected and they are connected by means of the tubules. Vesicles are small spherical structures which are cut off by this endoplasmic reticulum. Now, refinery, processing unit of the cell, middleman of the cell. What are its function? The name is indicative, same are its function, but before that it has two phases. The polarization is there. The stacks are arranged in such a way that it is having one convex surface and one concave surface. Now this convex surface is cis phase and the concave surface is the trans phase. The cis phase is generally towards the endoplasmic reticulum and the trans phase is generally away from the endoplasmic reticulum. It may head towards the plasma membrane or the cell membrane. The whatever vesicles are cut from the endoplasmic reticulum, they are coming and joining the Golgi complex and on the cis phase. So cis phase is the receiving phase. This is receiving phase. Cis phase of the Golgi complex is the receiving phase, and this trans phase is the shipping phase. Trans phase is the shipping phase. This one is shipping phase. Cis phase is the receiving phase. The membrane and the material comes from the endoplasmic reticulum to the Golgi complex towards this cis phase. It receives the membrane there. The vesicles are cut off from each of the cisternae and material goes ahead and then it reaches the trans phase. Now coming to the function. A variety of functions are done by this Golgi complex. The most important function, it is engaged in synthesis. It is engaged in synthesis of the proteins. It is engaged in synthesis of the lipids. Now we have talked about protein synthesis in endoplasmic reticulum. The proteins actually are synthesized by the ribosomes which are present on the outer surface of the endoplasmic reticulum, granular endoplasmic reticulum. These proteins then enter into the endoplasmic reticulum. They are initially linear, but they fold and they take a particular shape, their own shape, and then if they are secretory proteins, they are attached with some oligosaccharides. They become the glycoproteins. So glycoproteins are produced, synthesized, assembled inside the endoplasmic reticulum. Now see, whatever oligosaccharides are attached to this protein to form them glycoproteins, all of them oligosaccharides are generally similar. So all types of glycoproteins which are synthesized in endoplasmic reticulum, they are similar. They are not diverse. Now the diversification is done in the Golgi complex. When these glycoproteins, they are cut off in the form of vesicles, the glycoproteins in the lumen of these vesicles, such vesicles, transportation vesicles, are cut off from the endoplasmic reticulum. The vesicles come to the cis phase of the Golgi complex. They are received there. Now these oligosaccharides which are present in the glycoproteins, which are similar, now they are replaced by certain other oligosaccharides, some other monomers of the sugar molecules, polysaccharide molecules, and they are made diverse. Now whatever processing is done inside the Golgi complex, it is a step-by-step process. The materials come in the cis phase, in the cisternae towards the cis phase, and then in each of the cisternae, the enzymes are differing, different enzymes are present. So partially gradually they are processed. Some processing is done in the first cisternae, then some in the second cisternae, then in the third cisternae. Likewise, the material is sent ahead and ahead in the Golgi complex, and in each cisternae some processing, some refining takes place, and lastly when the material when the proteins or any material, lipids, when they reach toward the trans phase, they are totally processed material. The material is totally processed and now it is ready for export. It is ready for shipping, and that's why this is called as shipping phase. So gradual transformation of the material, gradual processing of the material takes place through this Golgi complex, and in this way the glycoproteins which were similar while entering into the Golgi complex, now they become diverse glycoproteins, different types of glycoproteins. The monomers of the sugars have been changed to bring the diversity among them. They are now having some recognition sites, and according to their recognition sites, the vesicles are created. Transportation vesicles are cut off from the trans phase of this Golgi complex, and then the vesicles go to their destination. They may go to the lysosomes, they may go to the plasma membrane or the cell membrane to export that material outside the cell, whatever its fate is, whatever its destination is. If the material is required by some other cell organelles, then it is given to that. For instance, some enzymes, digestive enzymes, respiratory enzymes are needed in the mitochondria, so material is sent to the mitochondria. So anyhow, the material is sent to its destination in the form of transport vesicles, and these transport vesicles are cut off at this trans phase. Now this synthesis of the protein, synthesis of the glycolipids, lipids, phospholipids, all types of lipids, they are synthesized partially in the endoplasmic reticulum, lipids especially in the SER, smooth endoplasmic reticulum, and then they are brought to the Golgi complex, and in the Golgi complex further the synthesis or the processing takes place. So synthesis, it also synthesizes some of its own materials also. It is not always the case that it processes the material of endoplasmic reticulum. Many times there are certain substances which are synthesized inside the Golgi complex also. Now this, along with synthesis, it is the other functions are similar to the endoplasmic reticulum. It is a part of GERL system where there is membrane flow. It is giving it is affecting the intracellular transportation. The material is taken from one place to another place. It is the shipping center. It carries the material or sends the material to their destinations. It helps in formation of the lysosomes. Lysosomes, we know, they are the suicide bags of the cells. Digestive enzymes are present in them. So all these functions, they are done by this Golgi complex. They help in all these ways: exportation, export, shipping center, processing unit, and they are very effective in secreting the enzymes and all other materials, hormones, enzymes, whatever materials they are needed to be secreted outside the cell. The Golgi complex is doing that in coordination with the endoplasmic reticulum. Many things are synthesized in endoplasmic reticulum, come to the Golgi complex, and then Golgi complex distributes them. So this is the importance of Golgi complex. Now we would discuss one more cell organelle of the endomembrane system, and that is lysosome. So let's discuss about the lysosomes. These are the simple structures, such type of spherical tubules, vesicles. Lysosomes are just vesicular in nature, small vesicles or large vesicles depending on the number of enzymes which are present in them. Now this lysosome is discovered by Christian Dalvi in 1955, but for the same he got Nobel Prize in 1974, sorry, not 75, 1974, the Nobel Prize for discovery of the lysosome. See what is the importance of lysosomes. So Christian Dalvi found these or discovered these lysosomes while he was studying some nerve cells in the old. He found them in 1955 and he got Nobel Prize in 1974 for the same lysosomes. We all know that lysosomes are very famously called as suicide bags. Suicide bags of the cells, because they are having hydrolytic enzymes inside them, and these hydrolytic enzymes are so many types, different types of hydrolytic enzymes like proteases, lipases. These enzymes can digest the macromolecules, all types of macromolecules like the proteins, lipids, carbohydrates, even DNA, RNA, means nucleic acids also. They can digest the macromolecules like proteins, carbohydrates, lipids, fats, DNA, RNA, nucleic acid, anything. And that's why they are very important in the cells. We know that these hydrolytic enzymes which are present in the lysosomes, they may range up to 16 number or more than 60 hydrolytic enzymes. Different hydrolytic enzymes are present in the lysosome. There are different lysosomes in which different enzymes are present. It is not the case that all the enzymes, hydrolytic enzymes, should be present in one lysosome. So there might be lysosomes which digest the lipids. There might be different lysosomes in which the enzymes for digestion of the proteins are present. So depending on that, but at a time 60 or more than 60 enzymes can be present in the lysosomes. Now I have objection about this suicide bag. We cannot call them as suicide bags, though they are helping in autophagy. Because these enzymes which are present inside the lysosomes work in a acidic medium. So the pH should be 4 to 5. Hydrogen ion concentration. Now the hydrogen ion concentration or the pH inside this lysosome is maintained acidic pH, strong acidity is maintained there, and this is maintained by pouring or introducing the H positive ions inside the lysosomes. So lysosomes have acidic medium inside them, and therefore the enzymes which are present in the lysosomes, they are active in that medium only. What happens now? Why they are called as suicide bags? Because it is general consideration that if the lysosomes burst open, the hydrolytic or the digestive enzymes might digest our cell organelles, other cell organelles, and the cell whole cell. This is called as autolysis. The cell digests itself, the enzymes, but that is not happening. Autolysis is not possible generally in general conditions, because whatever enzymes are present inside the lysosome, they are active in acidic medium. That acidic medium is maintained inside this lysosome, not outside. So when this lysosome breaks open, the enzymes are released into the cytoplasm, and in the cytoplasm enzymes are not active because they are not getting that pH. The medium is neutral, cytoplasm is neutral, it is not acidic medium, and therefore the same enzymes are not digesting the cell. They are not digesting the cell organelles, other cell organelles in the cell. So autolysis is not very common. It may take place in certain cases where the cell content is less and the huge amount of lysosomes are produced, and by chance all of them burst at one time, all of them break at one time, so many enzymes with change in the pH. If so many lysosomes are broken and the whole medium or cytoplasm becomes acidic, then the enzymes become acidic, then autolysis is possible. Otherwise we are living normal life. Many times the injuries are seen in our body, in organisms, and even then within these injuries, even when the lysosomes are broken, they are not causing any damage or any substantial damage to us. So calling them suicide bag is not that much proper. Now coming to the function of lysosome. Lysosomes structurally are membrane bound sacs, ultra microscopic sacs, and that is very simple, so nothing to talk much about the structure of the lysosome. Functionally they are important because they are our scavengers. They are the means of autophagy. They are the garbage disposal system. These two terms are same. Garbage disposal system of our body. They are our protectors or defenders. Let's see one by one. Scavengers. They, whenever some unwanted material is present in our body, this term is similar to garbage disposal system. So whenever we have any unwanted inside our body, that is digested by adding that material into the these lysosomes. Whatever we want to destroy, anything which is unwanted with us, anything which is extra with us, that is put in the lysosome and it is digested. So they are the scavengers, they are cleaning our body, they are garbage disposal system of our body. Autophagy is also very helpful phenomenon to some extent. Helpful because whenever we want some unwanted material to remove from our body and use that material, use the content of that material for some other as building block molecules for some other materials. For example, suppose we have some extra proteins and unwanted proteins. The proteins are put in the lysosome, they are digested there, means they are brought to the amino acid level. When proteins are broken down, amino acids are formed. Now this stock of amino acid is utilized for other purposes. They are utilized to form other proteins which are needed. So in this way, autophagy is helping us. Whenever we need some material at some different place, some unwanted material is broken down into its smaller units, and then these smaller units are used as building block molecules for construction of some other material. So that is autophagy. Now you might be knowing that one Japanese scientist got Nobel Prize for autophagy concept, and he for the first time said that when we fast for continuous 30 days or 40 days, we can remove cancer, because in cancer also there are unwanted cells, and in that fasting stages the unwanted cells are broken down to use material of the cells for some other purposes. So whenever we want material for some other purposes, some of the unwanted or excessive extra cells or cell parts, they are broken down and the material is used elsewhere. So that is autophagy. Protection. Lysosomes helps to protect us because lysosomes have digestive enzymes. So when a bacteria or virus enters in our body, we put that bacterium in the lysosome and the bacteria is totally digested, because whatever needed to digest the bacteria, the lipid breaking enzymes are needed, protein breaking enzymes are needed, carbohydrate breaking enzymes are, all of them are present with the lysosome. So bacteria are such foreign bodies, foreign pathogens, they are destroyed or they are digested inside the lysosomes. Now these lysosomes are formed by Golgi complex and endoplasm. Basically, whatever enzymes are present in the lysosomes, they are formed by the proteins surrounding the endoplasmic reticulum. Then the enzymes, the proteins become the enzymes inside the endoplasmic reticulum, and the membrane, again membrane factory. Endoplasmic reticulum is the membrane factory. So the membrane of the lysosome and the enzymes both are secreted in the endoplasmic reticulum. Then it cuts off the vesicles, but prior to become independent lysosome, they are again processed by this Golgi complex, and then Golgi complex releases the lysosomes. The lysosomes show polymorphism also. There are primary, secondary, tertiary lysosomes. When the lysosome which is cut from this endoplasmic reticulum or the Golgi complex, it is a primary lysosome. When some material is added to it for its digestion, then it becomes secondary lysosome. It becomes phagocyte when some food is added to it for digestion. We know phagocytosis in amoeba. Amoeba digests its food by phagocytosis method, where the membrane cuts off a vesicle and takes the material inside. That phagocyte is nothing but it is a lysosome, because digestive enzymes, hydrolytic enzymes are present in that lysosome. So phagocytosis or the pinocytosis, all these are the lysosomes. Phagocyte or pinocyte vesicle, these are the lysosomes. So polymorphism is the primary, secondary, tertiary, like that the different types of lysosomes are present. Enzymes also differ in this lysosome. So this is about the lysosomes, their structure and function. Christian Titouvé got Nobel Prize in 1974 for discovery of the lysosome. Now I have some slides of the endoplasmic reticulum, Golgi complex and the lysosome, formation of the lysosome, etc. I would so this is a plant cell. I have a purpose to show this to you, because I want to show you presence of these cell organelles. Now this is here you can see all this is endoplasmic reticulum, granular endoplasmic reticulum. Here also granular endoplasmic reticulum. You can see granular endoplasmic reticulum here also. You can see the smooth endoplasmic reticulum here. This is the smooth endoplasmic reticulum. I would highlight it. So here you can see the smooth endoplasmic reticulum, and this is a rough endoplasmic reticulum. This is a rough endoplasmic reticulum. This is a rough so endoplasmic reticulum is fast inside the cell. It is generally in continuation with the cell. Now I would again point out this with the pen. Here you can see this one. The endoplasmic reticulum is in continuation with the nuclear envelope. Space of the endoplasmic reticulum is continued with the space or lumen of the once again see here, here, here. The space of the endoplasmic reticulum is in continuation with the space of the nuclear envelope. Granular and rough and smooth endoplasmic reticulum. The smooth endoplasmic reticulum is chiefly in the form of tubules, and the rough endoplasmic in the form of vesicles, so cisternae. So these are the cisternae. Now this is the Golgi complex. This Golgi complex is having cisternae, then tubules and vesicles. So it has two ends. Golgi complex, this end is the cis end and this end is the trans end. The cis end is towards the endoplasmic reticulum, so it receives these blue colored vesicles from this side or from this side. It receives the blue colored vesicles like this. They become the part of this Golgi complex, and then it cuts its own vesicles from this side. So it is cutting vesicles on the trans phase. Now once again come to the same slide. So you can see here with this laser point, here is the lysosome. So here is the lysosome. So such type of lysosome, here is the lysosome. Lysosomes are also present inside the cell. Now come to the second slide. Here you can see endoplasmic reticulum in detail. This is in continuation with the cell. Here it is in continuation with the nuclear envelope. This is granular and this is a rough endoplasmic reticulum. Endoplasmic reticulum is synthesizing proteins, or it is modifying the proteins synthesized by these granules. Granules are the ribosomes. Then it is cutting off vesicles, and this material is transported to the Golgi complex. Then in the Golgi complex, this is the Golgi complex, cis phase of the Golgi complex, receiving phase, and this is trans phase or the shipping side of the Golgi complex. It has transport vesicles. They come from the endoplasmic reticulum, and these transport vesicles on the trans phase, they have been cut from this Golgi complex. So Golgi complex is the middleman. It is the export unit or shipping unit, processing unit, refinery center, or the middleman. It is called as dictyosome in the plant. So this is the Golgi complex. Again we show one dimensional structure in figure, but this is also three dimensional structure. You can see here these are the cisternae of the Golgi complex here. Now you can see these are the cisternae. So this cisternae is continued, and it is like this. It is three-dimensional structure, membrane bound structure. Now coming to the formation of the lysosomes. This is the way of formation of the lysosomes. You can see this endoplasmic reticulum is cutting off the vesicle. This vesicle then goes to the Golgi complex. From Golgi complex, then the typical lysosome vesicle is cut. The enzymes and the membrane comes from this endoplasmic reticulum. It goes to the Golgi complex. Golgi complex processes and forms it a lysosome. This lysosome is primary lysosome. Then it again combines with some material here and it becomes a secondary lysosome. So in this way digestion is done here. This is one way where the food is digested, which is coming from the outside. This is another way of autophagy, where the cell want to remove this material from its cytoplasm, so it is put into the lysosome and here the material is digested. So in this way autophagy is effected, and this is phagocytosis. Along with that, these are the scavengers, or they are the garbage removal system of the cell. Now come to the endomembrane system, what we have talked about. This endomembrane system includes the cell organelles like the endoplasmic reticulum. It begins with nucleus, envelope of the nucleus, then endoplasmic reticulum, then Golgi complex and lysosomes. These are the vesicles, also called as lysosomes. So in this way the material is transported from one place to another place. It is intracellular transportation, one thing. Another, it is flow of the membrane. The membrane of this nuclear envelope might go to the membrane of the endoplasmic reticulum. The membrane of endoplasmic reticulum goes to the Golgi complex in the form of vesicles. The membrane of Golgi complex goes to the lysosome, and this lysosome, when goes outside, its membrane is incorporated into the membrane of the plasma membrane or cell membrane. It throws out the material and becomes a part of that cell membrane. So this is a reverse in autophagy. When we have seen autophagy, here you can see some material is taken inside, a vesicle is formed here, and that vesicle has the membrane which comes from the cell membrane. Now this membrane forms the lysosome, food vacuole, etc., like this. So membrane flow is a continuous process, and this is important because the materials are transported with this membrane flow. So in this way the endomembrane system is working. So friends, I think it is sufficient for today's discussion. We have discussed today about the endoplasmic Golgi complex, then endoplasmic lysosomes, and now this is the whole endomembrane system, how it works. I have explained it with the help of these slides. I think it is sufficient. Thank you very much.